Hi, everyone, and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely, and today we are joined by two guests, Esraa AboJasser and Dr. Lee Williams. Listeners, Esraa is Market and Applications Manager in Analytical Testing at Biotage, and we're excited to welcome Lee back from our previous episode. Lee is Director of R&D for Analytical Consumables and Applications at Biotage. And listeners, I recommend checking out episode 160 first just to get some general history and background about Biotage, because today we're going to talk more about the company and how the team there is supporting anti-doping testing and analysis, really focusing in on their North American market. So Esraa, Lee, welcome to the show today. How are you?
Hi, Marie. I'm doing great. Thanks for having me.
Hey, Marie. Nice to speak to you again. Yeah, doing good. Thanks.
Well, we're excited to have both of you with us, and we're looking forward to jumping into our questions. And I know we had a chance to get to know Lee a little bit better in our previous episode. Again, listeners, definitely check it out if you haven't had a chance yet. So we'll turn it over to you, Esraa, first. Can you start by telling us more about yourself and your role there at Biotage?
Yeah, sure. So as the Market and Applications Manager for our Analytical Testing market at Biotage, my role is to support our sample prep customers from the different clinical, forensic toxicology, pharma, bioanalytical labs, as well as the sports anti-doping testing labs. So that's whether it's by providing them with application and method development and optimization support through my team of applications in Charlotte, or learning their needs and current challenges and partnering closely with the various teams at Biotage to help in addressing those challenges. So I liaison with all the different teams at Biotage, from R&D, product management, our service and commercial teams. And it's such a diverse role on the day-to-day.
Well, Esraa, this sounds like a really interesting position. And I think science career paths are often pretty diverse in terms of how people get to these different positions. So can you share, what path did you take to get to your current position there at Biotage?
My background is in biochemistry with a broad experience in synthetic organic chemistry. My career started in technical product support, focusing on what you could consider the two really integral chemistry lab techniques, chromatography and evaporation. This developed into a full product management role where I work closely with R&D, product engineers, other scientists in operation. And I got more involved on the business aspect of it and helping with developing and executing commercial strategies and market campaigns. From there, I ventured further on the commercial side, working with a number of pharmaceutical, chemical, environmental, food, and agriculture testing labs all over the Northeast and the United States. It was really an immersive experience in my career. It gave me exposure to a myriad of different lab techniques and technologies, how well they did in doing the job that they were designed to do. So when I came across this opportunity at Biotage, it was really the perfect fit with the skills and experience I brought to the table and the exciting multifaceted aspects of the position.
Oh, that's awesome. I like to hear about this evolution of your career over time. And we'll dive into more of what you're doing at Biotage. I guess, what do you think makes Biotage and its products different from other companies, perhaps different from some of the other groups that you've worked with previously?
I think our solutions are deeply rooted in strong expertise in purification and separation science. So unlike companies that may focus on one technology or become a generic supplier of multiple lab products, we focus on what many consider the most important, yet sometimes overlooked factor in analytical testing, which is sample prep. And we offer labs an integrated sample prep workflow solution with the dedicated application support. Biotage has long pioneered a diverse portfolio of advanced sorbents, polymers, and specialized chemistries. And today we continue to refine these technologies and innovate in this space. Another key aspect is that the innovation process at Biotage is really directly driven by the voice and the feedback of our customers. So we continue to focus on developing high quality solutions that are robust and reliable, workflows that streamline the sample prep process for analysts, reducing any process bottlenecks, and helping them save time.
I love that. And Lee, is there anything that you would like to add in terms of what really makes Biotage unique?
I think I'd agree with all of that. Where maybe we differ from some of the other manufacturers, we don't provide endpoint detectors like mass spectrometers. So the solutions that we provide really are from starting off with a sample right the way through the process in the analyte extraction, evaporation. So then everything associated with that workflow comes through to what then goes on to the mass spec, whether it's a GC or an LCMS. And I think it's because we've always been associated around the purification, the extraction. I think we've been able to help customers from that niche a lot more than maybe some other areas.
Oh, that makes sense. And I know in our previous episode, we really dove into the EMEA or Europe, Middle East and Africa region. And we're going to talk more about what's going on in North America with the two of you today. So let's dive into the details here. What does Biotage's footprint really look like in North America, Esraa, and who do you serve in this region?
Biotage has a significant footprint in North America, really strong direct presence in both the United States and Canada. Our operations are headquartered in our Charlotte, North Carolina office, where we serve there as a central hub for the Americas business. We also have two other key locations in Salem, New Hampshire and San Jose, California, which help us effectively serve customers both on the East and the West Coast. And for Latin America, we partner with a network of specialized distributors to make sure we have broad coverage. Our reach extends to different labs spanning different industries, from the environmental, water, agriculture, and food testing labs to the clinical forensic toxicology, and of course the anti-doping labs, all of which rely on Biotage's sample prep solutions for analyzing a wide range of different compounds and different matrices. Where you'll also see the full scope of our solutions is within the pharma and biopharma sector, where in early discovery labs will utilize our solutions for the synthesis and purification of both small and large molecule drug candidates. And as the drug candidates move into the late stage discovery and development, our sample prep workflows become then vital for bioanalytical workflows. And then we also work with the production phase in pharma. Our large-scale purification solutions serve both the small and the biomolecules. And beyond that, we also collaborate closely with academic and research institutes, helping them across a wide range of different research topics and disciplines.
That makes sense. Esraa, thanks for giving us a picture of what this footprint looks like. And it sounds pretty comprehensive. And I know different regions or maybe different markets have different needs oftentimes. So can you go into some detail on what are some of Biotage's biggest priorities for this North America region?
So what's really exciting about working in this North America region is that it's constantly changing. And as a result of that, our customer needs are also always evolving. Our priorities are always driven by those evolving needs of our customers. And many of the labs that we're working with are faced with similar challenges, I would say, when it comes to improving data quality, achieving reliable and reproducible results, while also trying to maintain cost effectiveness, efficiency, and productivity. For me and my team, one of our biggest priorities is application development and support. Whether it's the clinical diagnostic labs that need to make decisions for patient healthcare, or the forensic toxicology labs that are testing to inform criminal investigations and provide evidence, or the sports anti-doping testing labs that ensure that there's a clean athlete lineup for the different competitions. The analytical testing labs need optimized, validated methods that will consistently perform and produce accurate and reproducible results. So this is especially critical when it comes to supporting customers with those methods and helping them with developing those applications. And also it becomes really vital when you think about the new emerging analytes that add to already existing large panel of analytes, and as the sample matrices become more and more complex. So this is definitely a big priority for our team.
Oh, absolutely. And I think this is one of the things that makes it really exciting to work in anti-doping, is that things are constantly changing. New things are coming on the market, so to speak, in the therapeutic realm, as well as, unfortunately, in the doping realm. So maybe could you go into a little bit of detail about how that factors into the priorities?
We're constantly trying to keep up with the different new emerging compounds, understanding those compounds in terms of their chemical properties and choosing the right techniques to help extract them from the various matrices. Also, handling the sample matrices requires the method development and optimization. So this is what keeps our applications team constantly busy with all these different applications that emerge. What's really exciting is that we learn from one area and it helps us develop in another. So it pays to be involved in all those different industries and different applications.
Absolutely. I think anti-doping can learn a lot from the clinical, the forensic toxicology, all of these different worlds and vice versa. So I think that's great that you have a presence across the board. But let's talk maybe a little bit more in detail about the specific activities that Biotage has been involved with in North America. You mentioned in a lot of cases, you're working directly with customers, trying to develop these cool new solutions. Can you share maybe an example or two of some of these activities?
The activities really throughout the year will be different from technical seminars or webinars that we host or the user group meetings. We also participate at a number of different industry conferences, and we work closely with labs across the region on product development and product launches. One great example would be how busy it's been this summer for us with 2 critical product launches. Just a few weeks ago, we launched our Biotage Prep Expert 8, which is a fully automated sample prep system designed to streamline high throughput workflows in the environmental and PFAS analysis. And earlier in the summer, we also launched a novel SPE chemistry for the bioanalysis in oligonucleotides. So it's been exciting working with the different labs across the industry and seeing how those solutions work for them.
That's amazing. And Lee, is there anything that you would like to add about any of these recent activities?
The main thing probably to touch on is we're mass spectrometry based from a lot of the background, whether that's a forensic, environmental. One thing that was quite a challenge was certainly the development of the oligonucleotide product. That was heavily challenging for a variety of factors, a different chromatographic technique. So we didn't have the bandwidth to put a single LCMS system on using ion pairs. So we developed a helic chromatography separation, and then the different types of oligonucleotides you could be dealing with, different conjugations. Pretty challenging. Took up a lot of the summer.
Well, it sounds like both of you have been quite busy, and Lee, anti-doping labs specifically use a range of different consumable products for the routine work that they're doing every day, as well as the anti-doping research and development that they're contributing to. So Lee, can you go into some detail about Biotage's Evolute and Isolute series of columns and plates for solid phase extraction that you provide?
The one thing to take into consideration is there isn't a one size fits all. So both of these product lines are SPE based, so solid phase extraction. The Isolute is a silica-based sorbent, whereas the Evolute is polymeric. So that's based on a polystyrene divinyl benzene type copolymer. The silica tends to be slightly older technology. It's still widely applicable, heavily used in anti-doping and a lot of forensic tox labs. The newer polymers kind of came in more as a more attentive option to some of the C18 options from a silica modification. So when you're dealing with polar drugs, you get that enhanced retention mechanism. I think the other main thing is some of these polymeric materials are a lot more robust. So old school silicas tend to suffer from potentially phase collapse, drying out, reproducibility. Obviously, type 2 silicas were a lot better, and you're adding different modifications in. So they are actually better than the originals. But polymeric materials allow you to be a bit more, I don't want to say heavy-handed, but they're certainly more robust and can take a lot more abuse during your processing, whether that is the pressure that you're processing at, the pH range you're using, so various different things. Both product lines have a similar sort of sorbent modifications. So for different drug classes, you have anion exchange, cation exchange for both chemistries. In all honesty, they both work for many of these applications. So it could be quite simply down to personal preference with what lab uses what.
Well, that makes sense. And there's also the Isolute SLE plus supported liquid extraction plates and columns. So Lee, can you go into some details about how these products work and how they might streamline sample preparation in a lab?
So the SLE is very different to SPE. It is a plate or column-based approach, but the extraction mechanism is more based towards the traditional liquid-liquid type extraction. So for anyone dealing with liquid-liquid, it's kind of the same partitioning. So you're taking your aqueous matrix, you're mixing that with a water-immiscible organic solvent, and then looking for partitioning based on solubility, based on polarity. So log Ps, pKs are really important. Same processing parameters in terms of what's driving the extraction. But instead of the extraction interface taking place with two liquids in direct contact, the extraction interface takes place on a modified form of diatomaceous earth. So these materials have been around for many, many years, thousands of years. Mm-hmm. I think they're very highly refined. We do a lot of in-house processing. So they're not simply just dug out of the ground and put into columns, as some would believe. So we've got a range of specs for all of these materials. It does work differently to SPE. I think the main thing to take into account, SPE is based on an interaction. So you've got a material which may have a surface area for interaction of 500 to 600 meters square per gram. Diatomaceous earth products are typically less than 10 meters square per gram. So they're a low surface area. They work by providing a hydrophobized surface. So the water droplets go onto the phase. Processing is different because it doesn't actually have any flow through like you'd expect with SPE. So SPE, you'd condition the phase, you'd equilibrate, you'd load your sample, you'd wash and you'd dilute. The way SLE works, it's more of a hold-up technique. So you'd apply your aqueous sample that would absorb across the surface. There'd be no flow through. Simply wait 5 minutes and then apply your water-immiscible organic solvent. That partitioning then occurs during flow through. So it's a lot easier. Method development is generally a lot less involved. You don't get interaction in pores as you would with SPE. So really it's about trying to get your analytes in a neutral form where they become less polar and more amenable to extract into the water-immiscible organic solvent. It's pretty versatile. You can use it for acids, bases, neutral drugs. Neutrals tend to fall in. We've used it on a whole host of drugs of abuse, prescription medication, illicit drugs. Steroid hormone is probably the biggest application area within the clinical field. So anabolic steroids, they're pretty widely used. I think really the selectivity is brought by the way you use the product, as the technique is relatively non-selective. The selectivity and the extract cleanliness is really brought through the solvents that you use and your pH control and so on and so forth.
That makes sense. And I know, Lee, you work on the R&D side of things. And some people out there may think, okay, you develop a plate, you develop a column, you find something that works and the work stops there. Is that the case at Biotage, or do you keep developing new iterations and improving these products?
Everything's a constant improvement, I think. We're ISO registered. So by the very nature, there's a culture of constant improvement. It could be from a material route. So a lot of these chemistries have been around for many, many years. There's nothing new in a lot of the old silica type chemistries. We may refine and look at the sustainability aspect in terms of, okay, can we get to the same endpoint with greener solvents or less solvent or whatever that may be, so that you're always looking to improve the products, the cleanliness, the range of analytes. So a lot of analytical support, application support. The SLE was launched in 2004 and probably hundreds of posters and applications over the years to support that product to show applicability in different arenas, clinical, forensics, some environmental type targets. Acrylamide was one. So yeah, it's always ongoing, I would say.
Very cool. Well, I appreciate you talking a little bit more about these consumable products. And Esraa, I know we talked about Biotage's involvement in prior Olympic and Paralympic Games, particularly Paris 2024 in our previous episode. And we can't ignore the fact that Los Angeles 2028 Games are on the horizon in North America, of course. So Esraa, how do you anticipate the Biotage will be contributing to help ensure clean competition at these LA 28 Games?
We're excited for LA 28. We are already working closely and in close partnership with the WADA accredited labs in North America that support not just when they host Olympic competitions here, but they support daily and seasonal other professional sport competitions. And we're looking forward to support the UCLA lab, much like we did with the French anti-doping lab at the Paris Games. I think that automating the sample prep or the sample extraction on the Esraa workstation at the French anti-doping lab played a really critical role for them in being able to support the testing and helping with getting fast turnaround on samples and reducing any risk of errors from manual processing and just helping overall with improving data precision and accuracy. The UCLA lab is really not new to sample prep automation. In fact, actually, Ellie Ahrens, the director of the lab, presented on the role of automation and its evolution at the UCLA lab just recently at the PCC conference in April. In addition to the Esraa, the lab already utilizes Biotage's TurboVab nitrogen generators, as well as a few different sample prep techniques, some of which Lee has spoken about, like our Isolute SLE Plus and our Evolute ABN SV chemistries. So I know that the lab has already been in preparation, working to be ready for LA28, and we're looking forward to supporting them pre and during the competition.
Very cool. So it sounds like the UCLA Olympic Lab is not a stranger to Biotage. You've been working with them for a number of years. Can you talk about what that relationship or partnership has looked like over the years and what it might look like over the coming years in preparation for the games?
Yeah, we've been working closely with the UCLA lab, with the lab director and some of the scientists there. And this work has preceded my time at Biotage, whether it's with developing the methods using Biotage's extraction techniques from the SBE consumables to the Isolute SLE techniques that Lee had talked about. And with automating those methods on the Esraa, utilizing the TurboVab to really speed up the evaporation. And we will continue to work with the lab in the future and in prepping for LA28 to make sure that they are ready to handle the number of samples, to handle the fast turnaround requirements, to get those samples tested. And we'll continue to support them with those techniques and these solutions from Biotage. And we're looking forward to continuing this partnership during the games and in the future.
Very cool. And I think being able to participate in these large sporting events is an exciting part about being at Biotage, but you also do some really cool educational endeavors. And you touched on this in a previous response and talking about just some of the activities you're involved with in North America. But let's talk about these educational endeavors in more detail here. Can you share what are these webinars, tip sheets, and other resources that Biotage creates and provides, and why is this so important for the company to kind of have as part of your portfolio of things that you do?
Our goal, not just to be a laboratory provider of different solutions and supplies, but also to be a scientific partner to our customers, providing them with the various educational and scientific resources that are necessary in order to ensure their success with our workflow solutions. So in addition to product-specific documentation and support material that you'll find on our website, you'll also find an online library of scientific resources in various formats, including our on-demand webinars and live ones as well, application notes, white papers, blogs, and a library of application notes focused on all types of different applications spanning the different industries we serve. Obviously, the analytes that are commonly tested in the anti-doping labs like steroids or other illicit or performance-enhancing drugs. There's also opportunities for in-person trainings during conferences and workshops and the user group meetings that we host. And we focus not just on explaining the how when it comes to how our products work, but also explaining the scientific principles behind a particular technique. This really helps our customers not only follow the protocol or the methodology, but also understand it, and it enables them to be able to troubleshoot and optimize to their needs.
Well, I love this emphasis on being a scientific partner to your customers, to the people that you serve, and just to the scientific community in general by providing these resources. I think that's wonderful. And we've talked about the great things that Biotage is doing right now, and of course, some of the history in the previous episode, but let's look towards the future next. Lee, can you share what are some of Biotage's goals that you are working towards in the next maybe 5 to 10 years?
I think obviously company growth is massively on the agenda from an internal perspective, really bringing some of our recent acquisitions to fruition. We've purchased a number of companies that are really helping our traction in the large molecule or biomolecule arena. So really from that perspective, we're looking on the collaboration and how we can push a lot of these materials, not just from the high level purification side, but also right round onto the sample prep side. So I think that's going to be quite exciting moving forward. There's going to be emphasis on peptides, oligonucleotides we touched on a little earlier. So yeah, I think it's going to be a big learning curve for us as well, trying to work with some of our colleagues. But I think it's definitely exciting.
Absolutely. And Esraa, is there anything you would like to add in terms of your perspective on the goals?
Yeah, I definitely want to emphasize Lee's point. We want to take advantage of what our different partners have to offer and ensure that we are able to support customers with some of these new and emerging applications, especially in the large molecule arena, where we see that not just in new drug therapeutics, but it makes its way into the clinical biomarker diagnostics and also some as we continue to see in the anti-doping testing. So I think we still have our work cut out for us to continue to develop those applications for our customers and to support them with their method development needs.
Certainly. I think there's plenty to keep you busy in the coming years for sure.
Sure.
And to return to this idea of partnership that we mentioned, I think this is really important for anti-doping, particularly because it's a smaller field. So can you share why it's so important for Biotage to work with scientists, research funding organizations, and other partners within the anti-doping field to really help support clean sport?
I think it's crucial to work with all the different stakeholders in supporting clean sports. And when you think about the effectiveness of anti-doping testing, it's dependent directly on continuous advancement in analytical science and technology. For us at Biotage, understanding the analytical challenges, the regulatory changes is what directly will guide our resources and efforts towards continuing to innovate the solutions that we have, as Lee mentioned, into more practical solutions that help address the needs of the labs and help them stay ahead of the curve.
Definitely. And I think the PCC brings a lot of these relevant partners together every 2 years for its PCC conference. And we just had one earlier this year in April, as we mentioned. Lee, Biotage was a sponsor at this year's conference, and we were so excited to have you join us at the event. So can you share what your experience was like attending the meeting?
I really enjoyed the PCC event. I tend to like the smaller conferences. It's always far more intimate. You get to speak to people. It tends to be a lot more relaxed. We've all been to big conferences where there are 5,000, 10,000 people, but trying to get that right balance of the size and the technical, I think the PCC has done really well. The talks were diverse, so there's a mix of information really focusing on the science, upcoming trends. Gene doping is definitely something that is of massive interest, is regulatory. Former athletes presented. You don't normally get that sort of interaction or vision. So yeah, it's definitely a conference I would love to attend again and well worthwhile.
Oh, wonderful. And of course, we would welcome you back. And Esraa, is there anything you would like to share about your experience at the PCC's recent conference?
It was really great. It was really eye-opening when it comes to the presentations that we got to listen in, covering all the incredible work that goes on behind the scenes to organize sports testing and the various challenges that the anti-doping world is faced with, from the testing labs, the national and international WADA partners and agencies, and to the athletes themselves. We found it to be very insightful and very grateful to the PCC as well as the MLB and USADA for inviting us.
Well, we appreciate Biotage's support of the conference. It was wonderful to have you there, and we appreciate you sharing your perspectives with attendees at your table and just mingling with the crew and talking about the work that you're doing, because I think there are so many important projects and tasks to solve in the world of anti-doping. And I think Biotage is working on some cool developments to help address them. So Lee, in your opinion, what is one problem that you're really excited about working on in anti-doping, or that you would perhaps like to work on in the future?
There's lots of stuff going on. The gene doping, as I mentioned, is something that I have a real interest for. I have absolutely no experience up to this point, but I think it's absolutely fascinating, the lengths and the extents that people go to. So like the designer drug market, you've got a lot of smart people doing the work to identify where things are going. But there's also on the other side of the coin, a lot of smart people trying to be that one step ahead to go and cheat the system. I've always been fascinated by that balance. I think gene doping is definitely interesting. The biomolecule stuff coming through, whether it's peptide doping, is something that we will definitely be doing more work on. So yeah, it's a big learning curve.
Absolutely. I think it's an exciting time in the field, and we'll definitely be watching these particular areas. Esraa, to remind our listeners who might be looking for more in-depth information about some of the topics that we talked about today, where can they go to learn more about them and more about Biotage?
Yeah, so I recommend our listeners to visit our website, biotage.com, where, as I mentioned, they can find various educational resources on Biotage solutions and the applications they serve. I would also welcome our listeners to contact us directly. We'd love to hear from you.
Oh, wonderful. Well, we appreciate both of you sharing your perspectives. Listeners, definitely get in touch if you have particular questions or if you just want to learn more about the work that they're doing at Biotage. And Lee, Esraa, thank you for joining me on the show today. It was such a pleasure to speak with you both.
Thanks, Marie. It was a pleasure speaking with you.
Thanks, Marie. Thanks for having us back. It's been really good to spend time and discuss some of these things. And thanks to all the listeners.
Well, it was wonderful to chat with both of you. And listeners, it's always great to have you here with us as well. Please take a moment to subscribe to the show on your favorite podcast platform. And we hope you'll join us again for our next episode of the Anti-Doping Podcast.